Hello everyone and welcome to the next talk in the Eric Northeast and Tees Valley Wildlife Recorders Conference 2020. We are really pleased to have Dr. Martyn Kelly with us today talking about the hidden world of freshwater algae. So hello Martyn, and how are you?
I'm very well, thank you.
Good, yeah. Excellent, thanks. So I'm really looking forward to this talk. I did a little bit of ecology during my degree and I just found it absolutely fascinating. So it's just amazing to think of that sort of hidden world out there. So we're looking forward to getting some of your insights into that microscopic world. And so throughout this premiere broadcast, Martyn will be present and we can take questions in the chat or the comments section. And so just pop any questions or comments through there. We'll have contact details if you've got anything, you know, after the premiere and have got questions, we try and follow them up. And we like to hear from people. So Martyn, if you could just do a little introduction to yourself and just when you get started, just do your screen sharing. I'll mute myself and switch my video off and just enjoy your talk. So over to you, Martyn. Thanks.
Okay, thank you very much, Fiona. So, as Fiona said, I've got an interest in the freshwater algae. It's largely a professional interest. I'm an environmental consultant. I'm also an honorary professor at Nottingham University, but it extends into my my leisure time as well, and it's the area of natural history where I sort of spend most of my time and focus most of my attention. I write a blog which is supposed to be accessible for the layperson. It's called Microscopes and Monsters, and in this blog I talk about the sort of hidden biodiversity of the United Kingdom and beyond. So this is really the sorts of bugs and beasts that most people overlook most of the time. And that's called Microscopes and Monsters, and it's a WordPress. You can see its address at the bottom of the screen. So now we normally only notice algae when they become a nuisance, and you've probably seen signs like this beside lakes in the summer. Toxic blue-green algae, keep away from the water, don't let your dog in the water, no swimming, things like that. That's the sort of the public perception of algae, particularly freshwater algae a lot of the time, but they play many positive roles too. Algae actually constitute half of the United Kingdom's plant biodiversity. So there's sort of thousands of species, most of which are underreported and underrecorded, and that number's going up all the time, and I'll talk a little bit about that in a moment. They're the base of aquatic food chains. So midge larvae, mayfly larvae, caddisfly larvae would all be grazing on these, and these in turn will become the food for trout and other fish. So they're very important as part of the ecosystem, and also to support services, recreational services such as angling that lots of people engage with. Algae, through their photosynthesis, replenish the oxygen in streams. If you look closely at algal growth, you can often see air bubbles forming. As far as the algae are concerned, oxygen is one of its exhaust gases, something it wants to get rid of. But as far as the rest of the stream ecosystem is concerned, oxygen is a sort of vital resource. And they also play a role in stabilizing sediments and sort of creating ecosystems, which again, I'll mention that in a little while too. What are the algae? Let's ask ourselves this question before we get too deeply involved. Well, algae, they're simple plants. They're photosynthetic, means they're capturing energy from the sun and converting it into simple sugars. And they're not differentiated into roots, shoots, and leaves. That's sort of pretty much how most people would understand an alga. They also depend upon water, at least for the reproductive phases. So we can find algae outside the water. You might well find algae growing on a fence in your back garden, for example. But they will be periodically getting wet when it rains. And so there is always this sort of connection with the water. That's one of the shortcomings of the algal lifestyle. They're always dependent on water for certain parts of their life cycle. They're very, very diverse. We're talking about the algae as if they're a coherent group. They're not. They span 2 kingdoms. They include organisms that fit in the bacterial world. They include organisms that fit in the plant world. And they include organisms in 2 completely separate groups, the protista and the chromista, which are neither, strictly speaking, plants, and certainly not animals and not bacteria either. So they're very, very diverse. So it's not a coherent term, alga. It's a useful umbrella term for a large number of organisms that share some characteristics and habitats. But actually, when you get into the biology, they're very, very diverse and come from many different backgrounds. Now, one of the ideas behind this talk is to sort of encourage people to take a closer look at the algae. So we'll be coming back to this at the end. But first of all, let's have a look at this sort of this scale, just to put the algae into context. So on the left-hand side, we've got 100 meters, and on the right-hand side, we've got 1 micrometer. 100 meters, we're sort of looking at landscapes. We zoom in a bit to sort of the 10-meter scale, we can look at trees and shrubs. Zoom in a bit more, 1 meter. We've got the larger grasses, some flowers, maybe less than 1 meter, 10 centimeters or 1 centimeter, lots of other flowers, and we're starting to see some of the larger mosses as well. And it's in this realm, sort of round about a few centimeters, that some of our larger algae can be found. So we can see them with the naked eye in some cases. We can see them with much more clearly if we have a hand lens. So let's leave centimeters behind and move into millimeters. So we're straining now. We can still use our hand lens and just about detect some green specks maybe. You get a little bit smaller, so 1/10 of a millimeter, and now we're no longer thinking in terms of millimeters, we have to think in terms of micrometers. And from this point onwards, we really need a microscope. I'll talk a bit more about microscopes a little bit later. Some of the larger algae, sort of the desmids, you could see with quite a low power objective. We'll talk about desmids in a little while, but then we move on to the diatoms and the smaller algae, for which you need quite high power objectives. And just to make this talk as topical as possible, if you go a little bit to the right of the right-hand side of this scale, you get to COVID-19, which you can't see with a light microscope. You can just about see it with an electron microscope. So on the left-hand side in this picture, you can see my very first microscope, which I got as a Christmas present when I was about 12 or 13. In retrospect, I didn't see a great deal with it, and the optics weren't particularly good, but it did get me fascinated in freshwater life. And on the right-hand side, you've got the microscope I use at the moment for my professional work. And this one probably costs as much as a cheap car, I have to say, and this isn't the most expensive microscope that you can buy either. So when you're into professional microscopes, you are talking a lot of money. But as you'll see later, you can get started on microscopes for the price of a good camera. So I'm going to talk about 5 main groups of freshwater algae. Blue-green algae, which are the cyanobacteria, so these belong in the bacterial world. The green algae, which are the ancestors of land plants. And then 3 groups that you might know less about: diatoms, the euglenophyites, and the red algae. Let's start with the blue-green algae. The photograph at the top here is Upper Teesdale. It's one of the sites draining into Calgreen Reservoir. And if you pick up one of the stones in this stream, sort of a pebble-sized stone, you'll see it's got some sort of dark brown, sort of spherical colonies on it. And this is one of the things I want to sort of make you aware of when you're looking at the algal world. Sometimes it's quite nondescript specks and blobs. They're not necessarily green. They're sometimes brown. Sometimes they don't even look like living matter. But these, in this particular case, are colonies of a blue-green alga called Rivularia. It grows in Upper Teesdale and a few other remote parts of the country. And very simple, it's just a simple filament of cells. They gradually get narrower as you move towards the apex. At the base of each one, you get this sort of round cell, which is a heterocyst, and which is responsible for nitrogen fixation. So it can trap nitrogen out of the atmosphere and convert it into, well, ultimately convert it into proteins. So you can just about see the sort of the blue-greeny color in those cells, but when you look at it with the naked eye or a hand lens, you can see it's got this brown color. The reason for this, you can just about see the sort of the brownish hue in this photograph, is that they produce pigments outside of the cells, which act as a sort of sunscreen. This stone, I mean, to say sunshine in upper T cell might bring a wry smile to many of your faces, but nonetheless, these algae are exposed to quite a lot of sunshine, and just like us, would need to put some sunscreen on. And so over the years, over the millennia, algae have evolved sort of brown pigments that serve this sort of protective role. Here's another blue-green alga that you might have noticed on a gravel drive, for example. You might regard it as a bit of a nuisance. You get these sort of jelly-like growths, and it is in fact. Put it under the microscope, you don't need a particularly powerful microscope, and you can see it resolves into sort of these rosary-like chains, lots of small cells and one occasional bigger cells. The bigger cells again are these heterocysts which do nitrogen fixation, and it grows on freely drained environments such as this. And over the years, if you don't rake it away, which sort of most homeowners and gardeners would be wanting to do, it would spread and start to bind those stones together, and then you might start finding mosses growing in amongst it. So you're looking here at in a gravel drive at the first stage of a succession, and well, most of us quite like the idea of ecological successions in the right time and place, and we would regard our driveway as not being the right time or place for a succession, so we do our best to get rid of it. But that's what's happening on our driveways with this alga, which is called nostoc. Nostoc is pretty much the Latin for snot, if you're interested in such matters. Another sort of blue-green alga I want to talk about, this is from the upper part of the Ennerdale Valley in the Lake District. This is the River Liza, which flows into Ennerdale. I looked at a stone during the summer with some sort of greenish growths on it, but behind and underneath the greenish growths, this sort of dark olive green, sort of a mat. And that's another blue-green algae, This one's called Stygonema. And what we can see it doing in that mat again is forming a matrix, effectively forming a rooting substrate amongst which the green algae become entangled. Green algae, of course, don't have roots. They become entangled amongst these Stygonema filaments. So again, the Stygonema here was kicking off a successional process. And being a sort of a flashy Lake District stream, over time, these algae would probably get scoured away and the succession won't actually last very long. But this is sort of what we're seeing, the same processes that are happening on land, where you start with a bare piece of soil and end up with a forest. It's happening in streams, but those successions may be starting with blue-green algae or other very simple microorganisms heading into the algae, but then usually getting terminated by the vagaries of the British weather. So just a bit of background information on the blue-green algae. These are some of the oldest organisms known on Earth. We can find fossils of them that go back to the Precambrian. These were probably the organisms that first, were the first photosynthetic organisms, and these were the organisms responsible for pumping oxygen into the atmosphere and effectively creating the environment within which sort of subsequent organisms thrived. So although we often think of blue-green algae as a nuisance, they are actually extremely important in the story of life on Earth. I've mentioned already they're prokaryotes, not eukaryotes, they're bacteria, in other words. the cyanobacteria cells are the origin of the chloroplasts in other plant cells. So there's a process of symbiosis when cyanobacteria cells were engulfed by other organisms, and these, rather than being digested, the theory is these then became sort of co-opted as slave cells to do the photosynthesis, and it's from those everything that we now understand to be a plant has evolved. I've mentioned that some can fix nitrogen. I've mentioned also that some are toxic. So we owe an awful lot to the blue-green algae, and it's a little bit of a shame that most of us just think of them as being a nuisance. The next important group I want to talk about is the green algae. And this is a photograph of me in Upper Teesdale. I'm sort of crouched behind a boggy pool. I've taken a handful of sphagnum and I've squeezed it and collected the water that runs out of the sphagnum into a glass jar. And when I look at that under my microscope, there's a wealth of these amazingly shaped, usually symmetrical organisms called desmids. And all of these organisms came out of that one squeezing that I made in Upper Teesdale a few years ago. There's thousands of species of desmid. They're particularly associated with sort of boggy areas in upland UK. So as I said, you hunt out sort of boggy patches, usually with the more aquatic sphagnum, such as Sphagnum cuspidatum, the one that's supposed to look like a drowned kitten if you look at natural history literature. And you pull up a handful, squeeze it into your jar, and you should usually find sort of rich pickings of desmids there. So that's a different town. That one is in the Lake District near Coniston Water. And again, we found a wealth of different types of desmid in that sample. Green algae, much more various than just the desmids. I saw this patch of green on a stone submerged in Upper Tees, a little bit downstream from where I got my desmid sample. And I sort of stretched out and pulled a little bit up and put it in a bottle and got home. So there's a sort of macroscopic photograph of it just with an underwater camera. You can just see some narrow filaments. Pull the stone out, it's got a very sort of slimy feel to it. And put it under the microscope, you see these chains of cells, each of a single chloroplast. I was a little bit confused by these little growths on the side of it. Wasn't quite sure what they were. Worked out later, it's sort of one of the juvenile stages. So each of these cells eventually produces some spores. The spores, many of them will get washed away. Some of them will settle. Some of them seem to like settling on their parents' cells, from where they grow into another adult. And this is an alga called Uthric Sonata. And people have studied it over the years, and it's got quite a complicated life cycle with both asexual and sexual reproductive phases. One of the interesting things is this production of spores, or zoospores, called zoospores because they've got tiny flagellae. It happens when the days are short and the temperatures are low. So this is a species that actually thrives best in the winter. And that's the point I'd like to make to you with the microscopic world, that there's a lot going on, even in the depths of winter, and even when all of the higher plants seem to have shut up shop. So you can bring a sample back, you spend 5 or 10 minutes sampling, and I'm afraid it does involve putting your hand in some cold water sometimes. Once you've done that, you've got a sample you can bring back and look at it in the warmth of your house. So spend some evenings doing some microscopy and looking at the organisms I've been talking about. So there's some pros and cons to looking at the algae. There's that sort of little bit of cold phase when you're actually getting the sample, but then a lot what happens after that you can do in a nice warm centrally heated house. As another group of the green algae, you might be familiar with, because they're sometimes almost treated as honorary higher plants. So here's a little pond just south of Darlington called Croft Kettle. It's fed by a spring. It's actually on the magnesian limestone. And if you look in the water, it's very clear, and you can see lots of stems of Chara, like in those photographs. And they're often covered with sort of brownish growths of Diatoms, which is a group I'll talk about in a moment. The Chara are quite sort of stiff and brittle to the touch because they deposit calcium carbonate. You tend to find Chara in quite hard waters, and they sort of precipitate this calcium carbonate on their outside. This is a large and diverse group. The chlorophyta and the charophyta, we would collectively refer to them as a green algae, but they're actually two distinct groups. Some of these date from up to 600 million years old, which is the Cambrian era, so still a long, long time before the dinosaurs appeared on the scene. All of the higher plants ultimately evolved from these. We can find them in freshwater, marine and terrestrial environments. Planktonic, that means they're suspended in the water. Benthic means they live on the surface. Some of them are single cellular, but they extend right up to be to quite large organisms that may be a meter or more in length. The next group I want to talk about is called the Diatoms. This is a group I've made a special study of over the years. And these are some diatoms. Actually, I mentioned on the pictures of Chara, there are some sort of brownish diatom growths. And here's some close-ups of those diatoms growing on the stem of a Chara. And one of the things to notice about the diatoms is that they're not green. They've got this sort of yellow brownish colour. It's a sort of colour you would associate with seaweeds, wracks and kelps in the intertidal zone. And there is actually an affinity, strange as it may seem, between the diatoms and the brown algae. The most interesting thing about the diatoms, as far as identifying them is concerned, is they've got cell walls made of silica. That means they preserve in the sediments of a lake when they die. It also means they preserve on the slide very well as well. So when we study the diatoms, what we would tend to do is use oxidizing agents such as hydrogen peroxide to remove all the organic matter and then mount silica shells, which we call frustules, onto slides. And those slides, once you've done that, last a very long time. So this is a slide in my collection, which is over 100 years old. I was given it by an elderly diatomist just when I was starting out. And actually, interestingly, it also comes from Croft Kettle, and it's got a very similar assemblage of diatoms to those that we find there today. And we identify diatoms partly by the shape of these silica shells, partly also by the patterns, the ornamentation that you find on the outside of them. You see these lines here, which we call striae, this big distinct line going from one end to the other, which we call a raphe or raph. So this is, I think that's 25 micrometers long, so that's 1/40th of a millimeter, just to give you some idea of scale. And this is quite a large diatom. So with the diatoms, they're quite small. You need a fairly good microscope. Still, you can buy microscopes with which you can look at diatoms for, as I said, for the price of a decent camera. So a few more pictures of diatoms. This one is called Asterionella formosa, the beautiful star, if we translate the Latin. What's interesting, this lives in the plankton of lakes. What's interesting, all these dots along here, these are actually little fungi which are infesting the diatom, little fungi called chytrids. There's some of the wide range of shapes of diatoms that we can find in lakes and rivers. There are over 2,500 species of diatoms in UK and Irish fresh waters, all of them microscopic and endlessly fascinating. And one thing, oh, there's another one I'd forgot, a small diatom piggybacking on a larger diatom. But that leads me on. I said there's at least 2,500 species. One of the differences when you're studying the algae, studying the higher plants, is that you're much more likely to come across an organism that's new to science when you're exploring the microscopic world. So here's a stream that flows through Hampsterley Forest. Found a diatom, or we found some diatoms in a sample, took some photographs. made some investigations and found that nobody had described this before. So we actually named this. This is a species new to science that comes from Hampsterley Forest, and it's now formally described in the literature, and I sort of named it after my wife, gave it to her as a sort of birthday present. The diatoms, just to summarize then, very diverse group, over 100,000 species worldwide. Well, you see how the numbers are going up. I said 2,500 species 2 slides ago, and this one says 2,800, so probably be 3,000 by the time we get to the end of the talk. The earliest fossils are about 100 million years old, so the diatoms evolved round about the same time as the dinosaurs, let's just say, without getting too technical about it. Or probably the earliest mammals also from about the Cretaceous period as well. So towards the end of the dinosaur period, let's say. They've got this silica wall called a frustule, which makes it in some ways easy for us to study, and certainly easy for us to make permanent slides, which we can share with one another. There's a lot of this happened in the Victorian era, in fact, still does to some extent, that people would sort of post slides between each other and sort of compare the collections they'd made. There is still a postal microscopy society going in the UK, I think. So the yellow-brown colour I mentioned is due to fucoxanthin, which we can think of as a photosynthesis turbocharger. As diatoms can live in the depths of lakes where there really isn't much light, and we also find them to be quite prolific in the winter when again there's not so much sunlight coming through. So they've got this fucoxanthin as a sort of a turbocharger. Very important in the phytoplankton, sort of suspended in lakes, especially in spring. And I think in oceans, they're also extremely important. I think I'm right in saying that about 1/3 of all of the Earth's primary productivity is due to diatoms. So in other words, 1 in 3 of the breaths you take is oxygen produced by diatoms, which is quite a staggering thought. And the other thing is the diatoms which make submerged stones slippery. So if you've ever tried to walk across the stream and found your legs going in different directions, it's probably the diatoms you've got to blame for it. I'm back in Teesdale for this next slide, looking at a different group of algae called the Eucalyn Fighter. We were just walking along past Widlife Fell, going up the valley towards Cow Green Reservoir. This happens when you're out for a walk with me. I saw a sort of brownish film in a puddle and got excited and burrowed in my bag to see if I'd got a sample container. You can see it's not any old brown. That's a sort of a nice sort of gravy sort of brown. So I'm not just interested in sort of muddy browns, but I can detect a sort of a nice gravy brown from several meters distance and home in on it. This particular one was full of tadpoles. You see tadpoles, you think, well, they must be eating something. They won't grow into frogs unless they've got lots to eat. So I'd put a little bit of that brown film into my jar and put it under the microscope. And it's a group of algae called the Eucalyn Fighter, or more specifically, this one is called Phacus. This one, I think, looks a bit like an anarchist's bomb. If you think back to the sort of the old images of anarchists in the late 19th, early 20th century, with a sort of a sphere with, instead of a fuse, we've got a flagellum, which is a whip-like structure used for motility. So there you are, there's some algae just from a puddle beside the Pennine Way. And actually, if you look, there's an awful lot of euglenias described from puddles. They seem to like that particular habitat. So it's a much smaller group, the Eucalyn Fighters, and they're actually much closer to the animals. It's probably fair to describe the Eucalyn Fighter as animals with chloroplasts rather than as plants. Becomes quite a technical argument about exactly where they fit in the grand scheme of things, but Eucalyn Fighter are put there to sort of confuse people that want to make nice, neat divisions into plants and animals. Certainly, you look down a microscope, you see something that's not green and is moving around. It's quite a natural conclusion to think it's not a plant. And some of them even don't have chlorophyll, so sort of depend on getting their energy from outside sources. Heterotrophic would be the technical word for this. The final group I want to look at are called the rhodophyta, which are commonly called the red algae. Here's an example of a red alga, and it's certainly not red. I found this one in a small loch on Shetland, small freshwater loch. Put it under the microscope, you see this sort of beautiful appearance, this is central filaments with these sort of rosettes of side filaments coming off it. It's very slimy to the touch. It's often called the frog spawn alga, called Batrachospermum. And we find this in many parts of northeast England as well. Here's another red alga. You can find this one growing on stones in the faster parts of rivers throughout northeast England in the late winter, early spring. This particular stone comes from a river Wear or one of its tributaries, I can't exactly remember. And we've got these sort of wiry, hairy tufts of olive green filaments growing on the tops of rocks. Put them under the microscope, you see they've got a sort of a knobbly appearance. And this is a red alga called Lamania. Now, I have to admit to a certain little bit of weirdness at this point, because you can actually cook with Lamania. I read a paper a long while ago saying how they used it in parts of the Himalayas in cooking. So I've done some experiments. This is pasta alla carbonara with some Lamania added to it. I thought it was delicious. You have to ask my wife for a second opinion about cooking with freshwater algae though. So to summarize, the red algae, there's about 5,000 species worldwide, but most of them are marine seaweeds. So if you go to the seashore, look in a rock pool, you'll probably see some obviously red algae. And that's where most of the of this group of algae are found, in the marine, in the intertidal, in coral reefs. We do find some red algae that are red in rivers, but we also find organisms that are classified with the red algae, but which are not red as well, such as the Batrachospermum and the Lamania. So again, like we said for the diatoms, the red color is due to a sort of a turbocharger for the photosynthesis. In this case, it's a different one. This time it's a protein called phycoerythrin. They've got very complex life histories. I think I was almost put off algae when I was at university by people trying to explain the life histories of red algae to me as a first year. And again, in the freshwaters, this is a group that's most abundant in winter and early spring. So how do you get started with microscopy? Well, probably too much to compress into a end of a talk. So what I've done is there's a post on my blog which you can look at, which explains how to get started with microscopy. So just go to my blog, microscopesandmonsters.wordpress.com, type getting started with microscopy into the search box, and you should find the article which explains. And as I said, you can buy a reasonable priced microscope for the same price as a digital SLR. So they're not very cheap, but they're not unaffordable either. And once you've got it, there's all sorts of adventures you can have with it. The books you need to identify can be expensive, but there's an increasing number of online resources available as well. That's just a photograph of my own stack of books. I should add, not only are they expensive, but some of them, like these, happen to be in German, which doesn't help either. But as I said, there's some very accessible websites more available. This is one that I'm helping to produce called the Diatom Flora of Britain and Ireland. I talked about buying microscopes new, same price as a digital SLR, but you can also buy them secondhand as well. You could try places like eBay. There's also amateur microscopy groups such as Aquatic Microscopical Society, which has forums where you can sort of buy and sell equipment. So it's a good idea, actually. You can probably buy a better microscope for the same price if you're going second hand. I think in Victorian times, middle class Victorian families, a microscope probably played the same role as a television. You couldn't click the television on and watch a David Attenborough program. You had to get the microscope out and make your own adventures. And I really like to see more of that happening, people sort of discovering things and discovering the sorts of unimaginable worlds that you associate with a David Attenborough documentary, but finding those in ponds at the end of your garden. So that's all I've got to say, really. I end up with a photograph that I sort of summarizes, I think, my excitement with the freshwater algae. A mixture of what you can see here, some red algae, some blue-green algae, and some green algae, all growing side by side. I think if you looked in there as well, you'd find a lot of diatoms. So 4 completely different groups of organisms growing side by side in the middle of winter. And this particular stream is in Cumbria. So once you've got yourself sorted with a microscope, and once you've made a few forays and learnt where to look, it's a source of endless fascination. And I really do encourage you all to go out and have a go yourselves. Thank you.
That's Martyn. I don't know if you want to just yes, stop sharing your screen for a wee minute so we can see your lovely face again. That was absolutely fantastic. I suppose I'm asking the wrong person, but is it weird to feel so excited about algae?
I think if you define weird in strictly statistical terms, and on the basis that most people aren't fascinated by algae, then yes, it is weird. But it's also a case of
sorry.
It's also a case of most people not having the opportunities to get involved with the algae. And I think once you start looking at them, you do get a bit captivated, especially once you've looked at a few places and seen how there's sort of differences, let's say, as I was showing, between like a calcareous pond at one end of the Tees and an acid moorland pool at the other end of the Tees. So, and I think, well, it's a bit like all natural history. You get that initial trigger of interest, and then you start asking yourself questions, and then that triggers you to look at more places, and you find more differences, and you get a bit more interested. And that would apply whether you're looking at algae or mosses or higher plants or birds. So speaking strictly in statistical terms, yes, I'm weird, but I'm just a different sort of the general weirdness that is natural history. And looking in evolutionary terms, I would say it's the natural historians are the normal ones. It's the people that have lost the interest in spotting the organisms, you know, the differences, the organisms they can eat, the organisms they've got to avoid. I mean, they're the weird ones in an evolutionary sense, aren't they?
Absolutely, and count me in there. I know it's as well. There's the whole. It's just such a group of organisms, you know, with you know, being at the bottom chain, and also presumably with carbon capture, that they must be important for that as well.
Yeah, there have been some. About 15 years ago, there were some people saying there's all this, all these algae in the oceans, and maybe if we stimulated them a bit more, they could suck out a bit more carbon dioxide. And so there were big boats, you know, sort of research vessel size, going around sort of dumping iron filings in the ocean because they decided it was iron that the algae needed to grow more, in order to encourage the plankton to grow, in order to suck more carbon dioxide out of the atmosphere. That particular exercise, I think it was abandoned in the end, just being impractical. And I personally feel that you wouldn't destroy a fragile terrestrial habitat in that sort of cavalier fashion in order to capture carbon. And in a way, the oceans have got all this biodiversity, and if you dump fertilizer in it, you're effectively going to encourage the weeds to grow faster, and you're going to lose lots of the more sensitive organisms. So yeah, that particular exercise was sort of abandoned. I have to say, carbon capture is not really my my specialism. So I'm sure there's other people. They're certainly interested in seaweeds as biofuels. There's a lot of interest in that at the moment. It's a bit outside my specialism, I'm afraid.
Yeah, no, that's it's interesting to think about it. Yeah. Right, that's well, I absolutely loved that. That was excellent. So we'll just wrap things up there to say thanks so much for doing the talk. And hopefully people, as we said at the start, have been interacting in the chat area of the broadcast. And if you have got any questions, do just pop them through, and or get in touch afterwards, and we'll try and help you. And so thanks once again, Martyn. We really appreciate that, and absolutely loved that. Thank you.